Here I will cover some additional information about samples, specifically SARS-CoV-2 samples. So this is a slide that we saw earlier in the metadata model video, and you'll remember that I mentioned this ERC33 checklist. So this checklist is the checklist that we use for all viral pathogens, and I've included a link to this below the video. Feel free to visit this page and click around through the different fields, but as a quick overview, you'll find mandatory, recommended and optional fields in our metadata checklist. The mandatory ones, and in fact taxonomy is mandatory for every checklist and every sample. But in checklist 33, country is also mandatory, and some information about the hosts, a host sex, host scientific name, which in this case is almost always Homo sapiens, and an isolate name. Recommended fields include kind of more accurate geographic location, ideally latitude, longitude for most accurate, but region is very welcomed as well. Then things like host disease outcomes, so did the host recover, and isolation source host associated, which covers kind of where the swab may have been taken from, and other optional fields such as hospitalization status and sample storage conditions. So please feel free to take a look around the checklist and filter by these three conditions. So in some cases, mandatory field, perhaps you haven't collected it or you're not privy to this information. We do have a system for reporting missing metadata, which is INSDC approved, which means it's a system that's applicable not only within ENA but also NCBI and DDBJ. So we can use, let's say, not applicable if the information is not really appropriate to report, or not collected, not provided, to cover some other conditions. So while the fields are marked as mandatory, we do still allow this kind of missing value reporting. So in this workshop we will be submitting 3 example samples. These are prepared for you in the samples directory in the example data folder that you would have downloaded earlier. They share quite a lot of attributes, so obviously the taxonomy is the same. We've set collection date and country to be the same. So a lot of metadata is shared between these samples. I'll just note this sample capture status field. We strongly recommend that this is set to active surveillance in response to outbreak for all SARS-CoV-2 submissions, and that's really just kind of for tracking on our own side at the ENA. So these are the common attributes that these samples share. There are also some differences, so there are different sex and age, different disease statuses, so you'll see that sample 2 and 3 are diseased, and in sample 1 we've got one of the INSDC approved missing metadata fields, which is not collected. And then obviously they'll have to have different isolate names, and we've just generated some fake GISAID accession IDs for these as well. So we'll begin now by registering these samples, and later on we will submit some raw read data and some sequences against these samples.